In a remarkable scientific achievement, researchers at Harvard University have created the world's first menstruating mice. This milestone, led by Dr. Kara McKinley and her team at Harvard's Department of Stem Cell and Regenerative Biology, represents a pivotal shift in women's health research. For decades, menstruation has remained one of biology's most elusive processes, largely because the animals typically used in laboratory experiments do not menstruate. By bioengineering mice to naturally shed their uterine lining in a manner similar to humans, Harvard scientists have opened a new frontier in the study of reproductive health, promising to revolutionize our understanding of menstrual biology and related disorders. So, how did scientists at Harvard achieve this extraordinary feat? The team employed a chemogenetic technique, which is a method that enables precise control over specific cell behaviors using chemical signals. They introduced an engineered receptor into the uterine lining of female mice, allowing the endometrial cells to respond to calcium signals, a crucial factor in human menstruation. When these signals were paired with elevated levels of progesterone, the mice experienced what the researchers described as a controlled, reversible menstrual-like event that lasted several days. Analysis of the mice's uterine tissue confirmed the presence of bleeding, vascular expansion, and gene expression patterns that closely mirrored those observed in human menstrual fluid. Although the mice lack certain anatomical structures found in humans, such as spiral arteries, the overlap in gene expression reached 31%, a significant improvement over previous models. This breakthrough is far more than a technical achievement. It's a leap forward for scientific discovery. Until now, only a handful of species, humans, some primates, and a few bats, have naturally menstruated, making it extremely challenging to study menstrual biology in the lab. Dr. Kara McKinley, the study's lead author, explained that the absence of a reliable model had been a fundamental barrier in understanding menstrual processes. With this new mouse model, scientists can now investigate the full cycle of menstrual bleeding, repair, and regeneration. The research, published as a preprint on bioRxiv, also revealed that the uterus prepares for menstruation in a highly organized manner, with regenerating endometrial cells forming protective rings around clusters of degenerating cells. This intricate coordination helps minimize tissue damage during shedding. The implications of this discovery are profound. By providing an accurate laboratory model for menstruation, researchers can now explore the underlying causes of conditions like endometriosis, adenomyosis, and abnormal menstrual bleeding with unprecedented detail. These disorders affect millions of people worldwide, yet progress has been slow due to the lack of suitable animal models. The Harvard team's work offers a path forward, allowing scientists to test new therapies and interventions in a controlled environment. For example, the model could accelerate the development of targeted treatments, such as mRNA-based therapies for abnormal bleeding, which currently affects nearly 1 in 5 women. Existing treatments often rely on hormonal suppression or invasive surgery, but this new approach may pave the way for more precise and less disruptive options. Beyond basic research, the menstruating mouse model holds promise for translating discoveries into better care for patients. By replicating key aspects of human menstruation at the cellular and genetic level, scientists can now study why menstrual experiences vary so widely among individuals. This could help pinpoint the biological causes of severe symptoms, such as debilitating cramps or heavy bleeding, and lead to more effective, personalized therapies. The ability to test potential treatments in a realistic animal model is a crucial step in bringing new medications and interventions to clinical trials, ultimately improving the lives of those affected by menstrual disorders. This breakthrough is not just a triumph of biology. It's a symbolic victory for women's health research, an area that has historically been underfunded and overlooked. For much of scientific history, female animals were excluded from experiments to avoid the complexities of hormonal cycles, resulting in data that ignored half the population. Now, with a reliable model for studying menstruation, researchers can address long-standing gaps in knowledge and develop better treatments for everyone. While the findings await peer review, they mark a turning point in reproductive medicine, ensuring that women's health receives the scientific attention it has long deserved.